In a square dance, partners separate, then join with their corner for a dance around the square. The choreography of genetic inheritance has similar steps. The alleles of genes that govern a particular trait also separate and recombine during fertilization. This produces new genotypes in the offspring. Think of shirts in this dance as different alleles. When partners separate and recombine, the alleles form new combinations, just as meiosis and fertilization create new combinations of traits in the next generation. In this program, you'll see how the laws of genetics that apply to single-trait monohybrid crosses also apply to the inheritance of two traits, or dihybrid crosses. You'll continue to reflect on the work of Gregor Mendel, who pioneered genetic theory with his study of pea plants, and you'll learn how to determine the probability of phenotypes in human populations for two specific traits at a time, such as widow's peak and hitchhiker's thumb.
Now, you have a widow's peak. Do you know what that means?
Yes.
You don't? Scroll it back so it's here for me. Okay, now you got a widow's peak.
Why?
You've got a straight hairline. Some people have widow's peak, where the hair comes down to the point. Yeah, like that.
Okay, that's called a widow's peak.
Okay, you've got a straight hairline.
Excuse me, sir. Yes. Can I see your thumb for a second? It appears you have a curved thumb. Now, do you know what that means? It curves. It means you have a hitchhiker's thumb. Now, what that means is you've inherited two recessive genes from your parents. Now, when those two recessive genes come together, you get a recessive trait, i.e. the curved thumb.
If you could hold out one of your thumbs for us. Okay. You've got a straight thumb. And what that means is you received at least one dominant allele from one of your parents to give you the straight thumb. Got a straight.
Now, you got a straight thumb. Now, do you know what that means?
No.
That's good, because we can tell you. Now, that means a straight thumb is dominant. Now, what that means is you've inherited a dominant gene from one of your parents. Or, indeed, maybe even two dominant genes from both your parents.
Can you just hold out your thumb for us? We just need you to hold out your thumbs just so we can, just like this. Just so we can see your thumb. Okay, you've got straight thumbs.
Hold your thumbs up like this. Hold your thumbs like there.
Say cheese, you're on it.
So, are you ready for the data now, Nicole?
Yeah, Cam, but I still have some trouble with the symbols. Help me assign the alleles until I get the hang of it.
Okay. Now, individual number 1 has a widow's peak, which is dominant, so we can show that with a capital W. But because we don't know if the subject's other parent is dominant, which would be shown with a capital W, or recessive, which would be shown with a small w, we'll just leave it as a single dominant for now. Individual number 1 also has a normal thumb, which is dominant, so we can show that with a capital N. And the same rules apply as for the hairline, so we'll just leave it as a single dominant for right now. Individual number 2 also has a widow's peak.
Capital W because it's dominant.
Right. But individual number 2 has a hitchhiker's thumb. Now we know that a hitchhiker's thumb is homozygous recessive, so we can show that with two small n's. Individual number 3 has a straight hairline. Now, if a widow's peak is dominant and is shown with a capital W, then a straight hairline
shown by two small w's because it's homozygous recessive.
Right.
We've got the data and the final numbers for the 40 individuals we surveyed.
How many of them showed one recessive trait?
11 of them.
And how many showed two recessive traits?
Just 2. Is there a pattern there? I wonder if we looked at a third trait, we would find anyone who's recessive for all 3.
Well, there is a trait you could look at called attached earlobes. See how mine are attached?
Are they controlled by a single set of alleles too?
How would you determine that?
Well, let's see. We need a hypothesis, an if-then statement. If attached earlobes in humans were controlled by a single pair of alleles, and dominance occurred, then...
Then, when we sampled a population, only 2 possible results would be seen.
Yes. Earlobes can either be attached to the head region or not attached.
So what do you do now?
Well, we'll go out and sample a population and collect earlobe data.
And record the data. The number of people with attached earlobes versus those who don't.
Before you go, though, don't forget Nicole's original question, which was, do many people show several recessive traits?
Hi there. Could you just pull your earlobe out for us?
Sure.
I see you have a free earlobe. Now, what that means is from one of your parents you've inherited a dominant gene, because a free earlobe is dominant. Just pull your earlobe to the side for me.
It's gonna attach.
You got an attachment?
Good luck.
I see you've got a straight hairline. And can we see your thumb?
So she's got a straight thumb.
She's got a straight thumb.
Yeah.
Now can you just pull your earlobe to the side? And she's got an attached earlobe.
Yeah.
How did you do?
Well, we sampled 112 people. 31 had earlobes attached, and 81 had earlobes that were not attached. I think we can draw a conclusion and say our hypothesis was correct.
Why?
Well, we theorized that we would only find 2 phenotypes in the population, and our results clearly showed that. It appears that attached earlobes are recessive, but we'll have to do a little more research just to be sure.
Well done. Your information agrees with the research.
Well, that's good, because our population sample was pretty small. Larger numbers would have been more conclusive.
How did you get on with that question about recessive traits?
I checked that while we were sampling, but I didn't find a single person who had a hitchhiker's thumb, a straight hairline, and an attached earlobe.
I wonder why.
I asked similar questions myself, about 100 years ago. In my monastery garden, I studied the inheritance of pea plants for many years. I traced the offspring of pea plants that express 2 separate traits to determine the possible combinations of these traits. Here's a pea plant that has purple flower and long stem, 2 separate traits. In my day, I believed these traits to be controlled by specific factors. Today, you call these factors alleles. I performed a variety of crosses on pea plants and observed the offspring by germinating the seeds which the plants produced.
The reproductive structures of the pea plant are contained in each flower on the plant. They consist of stamens and the pistil. A flower self-pollinates when pollen from the stamens is transferred by wind or insects to the sticky surface on the pistil. A pollen tube develops and grows down the pistil to the egg cells in the ovary. The sperm nucleus in the pollen tube combines with and fertilizes the egg cell.
After these plants had self-fertilized, I examined the offspring. They all had purple flowers and tall stems, just like the parent plant. So the parent plant was true breeding for both these traits. I labeled the genetic factors capital P for purple flower and capital T for tall stem. This plant here is also true breeding, but it has white flowers and is dwarf. I knew from previous experiments that these traits were recessive, so I labeled their genetic factors with small p and small t. The next step was to combine these factors or cross-pollinate the plants. I wanted to know how the traits of flower color and stem length from very different parents would be expressed in the offspring.
pollen contains a sperm nucleus and the ovary contains an egg cell. The sperm nucleus and egg cell are gametes formed by meiosis. These gametes are haploid. They have only one set of chromosomes or genetic information, instead of the two sets present in other normal non-reproductive cells of the plant. Since each gamete can only carry one allele for any trait, the alleles must have segregated during meiosis, or gametogenesis. When the haploid sperm nucleus and egg cell meet during fertilization, the complementary alleles recombine as the diploid condition is restored. The result of fertilization is the creation of new combinations of alleles, genes, and chromosomes. What was Mendel about to do?
He was trying to cross pea plants with different colored flowers and different lengths of stems.
Right, a dihybrid cross. Now I have these giant chromosome models here to help you trace the travels of the alleles that Mendel identified. I've arranged them in a tetrad. Let's let these purple chromosomes represent the chromosomes carrying the gene for flower color, and these green chromosomes can carry the gene for stem length. I've arranged them in a tetrad because meiosis is the process by which parents produce the gametes. Can you show me the possible genotypes of the gametes produced by the parent?
Do you remember what the genotypes were of the plants that Mendel used?
Yes. The dominant one was all capitals. Big P, Big P, Big T, Big T. It had purple flowers and long stems. The other one was labeled with 2 little p's and 2 little t's. The recessive symbol for white flowers and a dwarf plant.
So if this was the chromosome that carried the gene for flower color, then we could label this allele as P.
Okay. Then we'll move the t's to the other pair of chromosomes, where the allele for stem length goes.
Good. Now the chromosomes have already duplicated, and they're lined up on the equator in tetrads in meiosis 1.
The first division separates the homologous chromosomes.
And each part receives a complete set of chromosomes.
Right. Haploid.
And then the chromatids would separate, and we would get 4 cells.
The end result is?
Gametes.
And what are the genotypes of the gametes?
Well, the genotypes are all the same. Big P, Big T.
We can show the other parent plant now. Use the same locations labeled flower color and stem length, but the alleles will be little p, little p, little t, little t.
We begin gametogenesis.
And the alleles separate.
The genotype of the new gametes are little p, little t.
And how many gametes would be produced?
There would be 4.
Right. But let's just leave these gametes here from the other parent, because I want to discuss fertilization. After fertilization, what are the genotypes and phenotypes in the first generation of offspring?
Well, we can show fertilization by bringing the gametes together. There are only 2 types of gametes. Big P, Big T, and little p, little t. When we match up the complementary alleles, we get the following genotype. Big P, Big T, little t.
The phenotypes for all the plants are the same. Purple flowers and long stems, just as Mendel recorded.
Not a single white flower or a dwarf plant. I didn't expect that.
I didn't either. And I didn't have models of chromosomes to help me figure it out. But why should I? I was discovering the laws of genetics. What I did is carefully pollinate pea plants and record my observations. From these observations, I made inferences of what I thought might be happening. Now, the next part in my studies was most important. This is what led me to explain the genetic factors you call alleles and the rules of inheritance derived from them. Here's what I did. I took plants from the F1 generation, the first filio generation, and cross-pollinated them. Now, the possible combinations of alleles was a little more complicated. Then, I just sat back and watched the next generation bloom. And what do you think I observed?
As a result of meiosis, each allele pair separates. We say that the individual alleles segregate. Mendel called this the principle of segregation. It became known as Mendel's first law. Later, by crossing heterozygous plants with more than one trait, Mendel discovered a very clear mathematical pattern in the phenotypes of the offspring. When plants of the F1 generation were crossed and their offspring, the F2 generation, were analyzed, Mendel realized that the segregation of one type of allele had not been linked to any other. They all segregated independently. This means that each offspring has a random chance of receiving any combination of traits. This segregation of alleles on different homologous chromosomes, independent of one another, was the only way that Mendel could explain the results of his F1 cross. It is called the law of independent assortment. If we simulate Mendel's F1 generation dihybrid cross, what will we find out?
The genotypes of the next generation, the F2s.
Which will tell us the phenotypes by looking at the dominant alleles.
And that should all follow the law of independent assortment. Let's let these two chromosome sets represent the chromosomes from the parent plants in the F1 generation. Now, if both of these are heterozygous plants, what are the possible genotypes of the gametes that each of these plants would produce?
Well, the alleles would separate, so any kind of P could go with any kind of T. That gives us 4 possible combinations from each parent. Capital P, capital T, capital P, small p, capital T, and finally, small p, small t.
Could we use our monohybrid Punnett square to predict the outcome of this dihybrid cross?
Well, we need a larger square to show all the gamete combinations.
It should be at least 4 by 4, because there's 4 different gamete combinations in every parent plant.
Here is the square. Go to it.
Okay, the gametes for the F1 generation go along the sides.
The gametes combine, and we can fill in the rest of the chart with the genotypes.
Out of 16 possibilities, we only get one plant that is a white flower dwarf specimen.
Oh, you sound surprised. I bet Mendel was too. What are your phenotypic ratios?
Well, we have 16 different combinations representing all the different genotypes. Some are heterozygous for one trait, homozygous dominant for one trait, or homozygous recessive for the other trait. And this verifies the law of independent assortment. One trait has no effect on the other. For example, the alleles for tallness don't affect the alleles for color. This verifies the law of independent assortment, that the alleles for each trait segregate independently during gamete formation and combine randomly during fertilization.
We have 9 phenotypes that are purple-flowered tall, 3 that are purple-flowered dwarf, 3 that are white-flowered tall, and 1 that is white-flowered dwarf. So we get a 9 to 3 to 3 to 1 phenotypic ratio.
Would you expect the same phenotypic ratio if you had a different dihybrid cross?
Yes, if both parents were heterozygous for both traits. You'd fill out the chart the exact same way, just changing the letters to represent whatever alleles you were trying to show. Now, isn't this the mathematical formula that Mendel noticed?
Yes, it is.
If one of the parents was homozygous for one of the traits, the ratio would change, wouldn't it?
Why don't you try?
Well, I have to change the...
These white songbird canaries are the result of the dihybrid cross of birds which carry the gene for white color and for song. It took many generations of breeding to produce this variety of bird, from the original waterslug or yellow canary with the beautiful song voice and a white, non-singing canary variety.
Oh, I just need a 4 to finish this game. Why can I never roll what I want?
Not lucky, I guess.
5. Well, ask me a science question from Chapter 5.
Okay.
Hang on, Nicole. I'll ask you a science question from Chapter 5. Why can't you ever get the roll that you want? This die has 6 sides. Now, what's the probability of my rolling a 3 on my next toss?
I'm not sure.
1/6.
Okay. And what's the probability that I won't roll a 3?
Well, there's 5 other numbers that could be rolled with just as much probability, so 5/6.
Okay, good. If the probability of rolling a 3 is 1/6, and the probability of not rolling a 3 is 5/6, then the probability of either of those two events happening is 1/6 plus 5/6, which equals?
1. The sum of all possible outcomes always equals 1.
Well, you know your math, Cam. Here's a little trickier question. What's the probability that the same number will come up on 2 different dice rolled at the same time? They're independent events, just like when the alleles separate into the gametes. Neither roll of the die influences the other roll.
How did I know this was going to end up as a genetics lesson?
Absolutely. Probability is a big factor in the phenotypic ratios we come up with. What's the probability that both dice will come up 6?
Well, I believed you when you said there's a 1/6 chance of any particular number coming up. So both would have a probability of 1/6.
Okay. Roll the dice 6 times.
6. You'd have to roll those dice for a long time in order to get 2 6s.
Well, it would be the same for any combination.
Exactly. There are actually 36 different combinations of numbers. We can figure out the probability that both dice will come up 6 by multiplying the independent probabilities that one die will come up 6. So 1/6 times 1/6 equals?
1/36.
Right. And as Nicole pointed out, that would be the same probability for any specific combination of numbers. It's called the product rule. And for genetics, it really helps make calculation for dihybrid and multihybrid crosses a lot easier. Remember this dihybrid example? The alleles from heterozygous plants segregate randomly. So there's a 50-50 chance, or 0.50 probability, that any allele will be found in any gamete. So that's a 0.5 probability. Let's determine the probability that the result of this cross will be a dwarf plant. That's this genotype here, small t, small t. First, we need to determine the probability that a small t will be in the ovum.
Well, that's 2 in 4, so 1/2.
Or 0.50. What's the probability that there will be a small t allele in the sperm?
Also half.
Or 0.50. Using the product rule, 0.50 times 0.50 equals 0.25. So we would predict that 1/4 of the progeny would be dwarf.
And there's 4 out of 16 that are dwarfed, which is 1/4. So we're right. We could just do 2 small squares, find the probability of a trait occurring in one square, and multiply it by the probability of a trait occurring in another square to find out the probability of both traits occurring together at the same time.
You want to try it again?
Actually, I think I can show you. Here's the dihybrid cross that we did. And in this Punnett square, we have the gametes for flower color. And in this Punnett square, we have the gametes for stem length. So we combine the two to show the genotypes of the possible offspring.
Okay, I see. So 1 in 4 plants will receive the recessive white flower, and 1 in 4 offspring will receive the recessive dwarf stem. And 1/4 times 1/4 is 1/16. We get the same results.
We were surprised by the results of our F1 generation cross. Mendel must have spent a long time working on his results.
I did, but it was the science of discovery that kept me working. You've only worked with dihybrid crosses. I worked with 7 different traits in pea plants and discovered that my laws of inheritance held true, as did the rules of probability. So, learn them well.
Dihybrid crosses can be treated as 2 monohybrid crosses. Using human traits, consider the monohybrid cross of parents heterozygous for the gene for earlobes. Free earlobes are controlled by the dominant allele, capital F, and attached earlobes by the recessive, small f. Then consider a monohybrid cross between 2 parents heterozygous for hitchhiker's thumb. The straight thumb is determined by the presence of the dominant allele, capital N. The bent thumb is due to the presence of only recessive alleles, small n. The monohybrids can now be combined to calculate the probabilities of a dihybrid cross in which both parents are heterozygous using the product rule. The probability of independent events occurring simultaneously is equal to the product of these events occurring separately. The probability of these heterozygous parents producing a child with attached earlobes and a straight thumb is 1/4 times 1/4, or 1/16. Can you predict the outcome of a cross between two individuals heterozygous for 3 traits? Widow's peak, hitchhiker's thumb, and free earlobes.
You could if you did a Punnett square of a trihybrid cross, but that would be kind of difficult to do.
Could you do it using 3 monohybrid crosses? Could you figure out the combinations that way?
Yes, if you used the product rule. You could multiply the probabilities of the monohybrid crosses in order to find out the trihybrid offspring.
Here you go, Nicole.
So, we can see that the probability of a person having all 3 recessive traits is 1/4 chance straight hairline, 1/4 chance hitchhiker's thumb, and 1/4 chance attached earlobe.
Using the product rule, 1/4 times 1/4 times 1/4 is a 1/64th probability that you'll receive all 3 recessive traits, assuming both parents are heterozygous for all 3 traits.
Excellent. From a few garden peas have come the universal laws that govern genetic inheritance. But humans are certainly not all peas from the same pod. In fact, with all our traits, a human gamete has the possibility of having over 8 million different combinations of alleles. The fertilization of 2 gametes resulted in this unique individual. But she is only one of a possible 64 trillion different babies her parents could have produced, based on the effects of the law of independent assortment alone. If we take into account the effects of crossing over and mutations, the possibility of variation increases. Gregor Mendel took the first steps in building the foundation for modern-day genetics. His laws are simple and elegant. They are the basis of all our understanding about inheritance. The law of segregation explains that alleles separate during gametogenesis and move independently of each other. The law of independent assortment states that each characteristic is inherited independently from all other inherited characteristics. And the product rule allows us to calculate the probabilities of multiple trait inheritance. These rules are fundamental to our understanding of genetics and the choreography of generations.